Welcome to the Deep Dive, your shortcut to being well-informed. Today, we're taking a fascinating plunge into how our muscles, these incredible machines, fuel everything from a blank to a marathon.
That's right.
Our source for this deep dive is a comprehensive chapter from Marks Basic Medical Biochemistry, and we're going to unpack it step by step. We'll focus on the core concepts, those biochemical pathways, and even some clinical insights.
Exactly. Our mission today is really to guide you through the intricate biochemical strategies our muscles use. We'll explore the different types of muscle, get a handle on the universal mechanics of how they contract, and then dive into their unique energy demands.
And how they adapt, right? Like resting versus going all out.
Precisely.
Yeah.
How those metabolic pathways shift, whether you're just sitting there, maybe facing starvation, or really pushing your limits during exercise. And we'll definitely highlight the clinical relevance, making these complex ideas hopefully really accessible.
Yeah, that's the goal. Make these intricate biochemical processes as clear as possible so you don't need the textbook right in front of you.
Yeah.
Just, lean back, listen, and hopefully get a feel for the incredible molecular ballet happening inside your own body. So let's start at the beginning. What are muscles, really? Your body has three main types, right? Skeletal, smooth, and cardiac.
Correct.
Skeletal muscles are the ones you consciously control, the ones moving your bones. Smooth muscles are kind of the quiet, involuntary workers in your organs.
Doing things automatically.
And then there's the cardiac muscle, just tirelessly pumping your heart.
And what's truly remarkable here is that despite these diverse functions, all muscle types actually share a fundamental mechanism for contraction.
Okay.
It's all based on an actin-myosin sliding filament system. It's beautifully orchestrated by changes in intracellular calcium levels.
Calcium, again, always key.
Always key. But while that core mechanism is universal, the strategies they use to fuel that contraction are quite distinct. They draw energy from a similar pool of primary sources: stored glycogen, circulating glucose, fatty acids, and sometimes even amino acids.
Okay, let's zoom in on skeletal muscle first, the ones we think about when we, lift weights or run. If you look at them under a microscope, they have that striated pattern, like stripes.
Yes, that's due to the highly organized arrangement of those actin and myosin filaments. These muscle fibers are long, cylindrical, and interestingly, they're multinucleated, almost like many cells fused together.
And what about the key structures inside? You mentioned the sarcolemma.
Right, sarcolemma is the outer cell membrane. Inside is the sarcoplasm, the intracellular fluid. And crucially, you have the sarcoplasmic reticulum. Think of it as the muscle cell's dedicated calcium storage tank. And then there are T-tubules. These are tiny tunnels, invaginations of the sarcolemma, that dive deep into the muscle fiber. They're essential for allowing the nerve signal, the action potential, to spread rapidly throughout the entire cell almost simultaneously.
That intricate setup sounds absolutely vital. What happens if something goes wrong there?
It is vital, absolutely. And we see its importance, sadly, highlighted in conditions like Duchenne and Becker muscular dystrophies.
Ah, yes.
These diseases stem from the absence or a mutation of a protein called dystrophin. Dystrophin is crucial for maintaining the structural integrity of that sarcolemma, the cell membrane. Without functional dystrophin, the membrane becomes fragile, it breaks down during contraction, leading to progressive muscle damage and loss of function. It really underscores how critical every component is.
That's a really powerful illustration. Now, even within skeletal muscle, we talk about different fiber types, slow twitch and fast twitch. What makes them so different? How does that affect what they do?
Yeah, it's all about specialization for different jobs. Slow twitch fibers, type Is, they're your endurance specialists.
Marathon runners.
Exactly. They're packed with mitochondria, it cells powerhouses, and lots of myoglobin, a protein that stores oxygen, which gives them a reddish color. That's why they're often called slow oxidative fibers. They rely heavily on oxygen-dependent or aerobic metabolism, making them incredibly resistant to fatigue. They develop force more slowly, but can sustain contractions for a very long time. Think of your postural muscles, like the soleus in your calf, working subtly all day to keep you upright.
Okay. And the fast twitch.
Fast twitch, or type 2, they're built for speed and power. They have fewer mitochondria, less myoglobin, so they look whiter. Type IAG, for example, are loaded with glycogen, they're stored glucose, and primarily use glycolysis, that rapid but less efficient anaerobic process for energy.
Quick bursts.
Quick bursts, exactly. They develop greater force much more quickly, essential for sudden, powerful movements like sprinting or lifting heavy weights. The trade-off, though, is they fatigue much faster, partly due to the buildup of lactic acid from that anaerobic glycolysis. There's also type Cias, which are sort of an intermediate blending characteristics of both.
So, Type I for the long haul. Type II for the explosive stuff. Got it. What about smooth muscle? Where do they fit in?
Smooth muscle is involuntary. You don't consciously control it. The cells are spindle-shaped, have a single nucleus, and, as the name suggests, they lack those striations we see in skeletal or cardiac muscle.
Right.
You find them lining the walls of internal organs, your digestive system, blood vessels, bladder, uterus. They're remarkable for their ability to maintain tension for extended periods using very little energy. Think about maintaining blood pressure or moving food along your gut. That's smooth muscle work.
And then the heart itself, cardiac muscle. Let's be unique.
Very unique. It's kind of a hybrid. It's involuntary, like smooth muscle, but it is striated, like skeletal muscle. The cells are sort of quadrangular and they're interconnected, forming a network. This allows them to contract synchronously as a single unit, a functional syncytium.
Team effort.
Absolutely. And the heart is the ultimate endurance machine, right? It's constantly pumping, so it relies almost entirely on aerobic metabolism. Cardiac cells are packed, absolutely teeming with mitochondria and have very little stored glycogen. This makes them extremely dependent on a constant oxygen supply.
Which is why heart attacks are so dangerous.
Precisely. In a myocardial infarction or heart attack, when blood flow and oxygen are cut off, glycolysis alone just cannot meet the massive energy demand. It highlights that critical reliance on oxygen.
It's fascinating how each type is so perfectly engineered. But, okay, how does any muscle actually get the signal to contract? You called it a biochemical dance earlier.
It really is. It all starts with a signal from a motor neuron. The point where the nerve meets the muscle fiber is called the neuromuscular junction.
Right.
Here, the nerve ending releases a chemical messenger, a neurotransmitter called acetylcholine.
Acetylcholine.
Right. Acetylcholine crosses that tiny gap and binds to specific receptors on the muscle membrane, the sarcolemma. This binding triggers the opening of sodium channels. Sodium ions rush into the cell, creating an electrical signal and action potential.
And then spreads.
It spreads rapidly along the sarcolemma and then, crucially, dives deep into the muscle fiber through those T-tubules we mentioned.
Ah, the tunnels.
Exactly. As the action potential travels down the T-tubules, it triggers the release of calcium from its storage site, the sarcoplasmic reticulum. This calcium is released through specialized channels called ryanodine receptors.
So calcium floods the cell interior.
It floods the sarcoplasm, and once it's there, calcium binds to a regulatory protein called troponin. This binding causes troponin to change shape, and in doing so, it pulls another protein, tropomyosin, away from the myosin binding sites on the actin filaments.
Clearing the way.
Clearing the way. With those sites now exposed, the myosin head, which is sort of primed with energy from ATP hydrolysis, can bind tightly to actin. Then it pivots, pulling the actin filament inwards. This is the power stroke that shortens the sarcomere, the basic contractile unit. That's the contraction.
And how does it let go to contract again?
Good question. For the myosin head to detach from actin, a new molecule of ATP must bind to it. Once ATP binds, the myosin head releases actin. The ATP is then hydrolyzed, re-energizing the myosin head, making it ready to bind again if calcium is still present. This cycle bind, pivot, detach, re-energize repeats as long as calcium levels remain high and ATP is available.
And relaxation.
Relaxation happens when the nerve signal stops. The calcium release channels close, and a very important energy-requiring pump called the sarcopump actively transports the calcium back into the sarcoplasmic reticulum. As calcium levels in the sarcoplasm drop, tropomyosin moves back to cover the myosin-binding sites on actin, and the muscle relaxes.
So this whole elegant biochemical dance, what does it mean for our health? This precision must be absolutely critical.
Couldn't be more critical. I mean, take ryanodine itself. It's named after the plant it comes from. But the toxin inhibits those calcium release channels. It acts as a paralytic agent. That tells you right there how fundamental that calcium surge is.
Wow.
Then think about acetylcholine. It needs to be cleared away quickly from the neuromuscular junction, otherwise the muscle would stay contracted. There's an enzyme called acetylcholine esterase that does this job, breaking down acetylcholine very rapidly.
So what if that enzyme is blocked?
That's exactly what happens with certain nerve gases. They inhibit acetylcholine esterase. Acetylcholine builds up, causing continuous muscle stimulation, leading to paralysis, respiratory failure, and it can be fatal.
Terrifying.
It is. But conversely, we use reversible inhibitors of acetylcholine esterase in medicine. They temporarily boost acetylcholine levels and are used to treat conditions like myasthenia gravis or even some symptoms of dementia. It just highlights the incredibly fine balance of these molecular controls. Life and death can hinge on these enzymes working correctly.
That's a stark reminder indeed. Okay, let's shift gears a bit and talk about fueling these incredible machines. We know muscles use glycolysis and burn fatty acids, but how is the regulation of these pathways specifically tailored for muscle?
Yeah, that's where muscle biochemistry gets really specialized. Let's take an enzyme involved in glycolysis regulation, phosphofructokinase 2, or PFK2. In your liver, when PFK2 gets phosphorylated, it usually slows down glycolysis.
Right.
But in skeletal muscle, phosphorylation doesn't inhibit PFK2. And even more interesting, in cardiac muscle, phosphorylation, for example, by insulin signaling or when energy is low via AMPK, actually activates PFK2.
So the heart can ramp up glucose use even when the liver is trying to conserve it.
Exactly. It's a crucial adaptation for an organ that absolutely cannot stop working and needs flexible fuel options.
Okay, what about burning fats? How is that regulated differently in muscle?
That's another elegant system. Muscle cells don't really synthesize fatty acids, but they have a specific enzyme, ACC2 acetyl coenzyme, a carboxylase 2, whose main job is to regulate fatty acid oxidation, or burning.
How does it do that?
ACC2 produces a molecule called malonyl-CoA, and malonyl-CoA acts as a potent inhibitor of CPTI, that's carnitine palmitoyl transferase I. CPTI is basically the gatekeeper enzyme that allows fatty acids to enter the mitochondria where they're burned for energy.
So ACC2 makes malonyl-CoA, which puts the brakes on fat burning.
Precisely. It's a way for the muscle to say, okay, we have enough energy maybe from glucose, let's not burn fat right now. But muscle also has another enzyme, malonyl-CoA decarboxylase, which is the opposite. It breaks down malonyl-CoA.
Releasing the brake.
Releasing the brake, exactly. This allows CPTI to work and promotes fatty acid oxidation. And the activities of these two enzymes, ACC2 and malonyl-CoA decarboxylase, are tightly controlled. For instance, when energy levels in the muscle cell are low, an important sensor kinase called AMPK becomes active.
Oh, me see.
AMPK inhibits ACC2, less brake, and activates malonyl-CoA decarboxylase, more brake release.
So low energy flips the switch to burn fat.
Flips the switch hard towards burning fat. It's a rapid and powerful way for muscle to prioritize fatty acid oxidation when energy is needed, especially important during exercise or fasting. Its fascinating studies in mice engineered to lack ACC2 showed they had significantly less body fat because their muscles were constantly burning fatty acids almost unregulated. It shows how critical this regulation point is.
That's incredible control.
Mhm.
So we know muscles need ATP constantly, but you said ATP itself isn't stored in large amounts. What does muscle use for those immediate, quick bursts of energy, like lifting something heavy suddenly?
That's where the creatine phosphate system shines. It's like an immediate high energy reserve. Creatine itself is synthesized mainly in the kidney and liver, then travels to tissues with high energy demands like muscle, brain, and heart.
Okay.
Once inside the muscle cell, an enzyme called creatine phosphokinase, or CPK, sometimes called CK, catalyzes a really important reversible reaction. It takes a phosphate group from ATP and attaches it to creatine, forming creatine phosphate and ADP,
Storing the energy.
storing that high-energy phosphate bond. Creatine phosphate levels in resting muscle are much higher than ATP levels. When the muscle suddenly needs a lot of ATP fast, like at the start of exercise, CPK rapidly reverses the reaction, taking the phosphate from creatine phosphate and adding it back to ADP to regenerate ATP instantly.
Like a quick charge battery.
Exactly like a quick charge battery right there in the muscle, ready to go. Creatine phosphate also seems to help shuttle energy from the mitochondria, where most ATP is made aerobically, out to the myosin filaments where it's actually used for contraction.
And what about creatinine? We hear about that on blood tests.
Right. Creatine and creatine phosphate aren't perfectly stable. They slowly, spontaneously cyclize to form creatinine. This creatinine isn't used by the body. It just diffuses out of the muscle into the blood and is then filtered out by the kidneys and excreted in the urine.
And the amount is constant.
Remarkably constant for an individual because it's produced non-enzymatically at a rate proportional to your total creatine phosphate pool, which is itself proportional to your muscle mass. This is why measuring creatinine levels in the blood, or creatinine clearance by the kidneys, is such a useful clinical indicator.
And here's where that seemingly academic detail has a very real-world impact for clinicians.
Absolutely. Let's take the case study mentioned. Renee F., a 9-year-old girl with acute post-streptococcal glomerulonephritis, or FLESGN.
Yeah.
Her blood test showed an elevated serum creatinine level 1.8 mg/dL, which is significantly high for a child her age. For a clinician, this immediately signals a problem. It indicates that her kidneys aren't filtering waste properly, their excretory function is decreased because the glomeruli, the tiny filtering units in the kidneys, are inflamed due to the recent strep infection. So measuring creatinine is a fundamental way we assess kidney function.
That makes sense.
And there's another muscle connection, too. If muscle cells get significantly damaged, say in trauma or certain diseases, they release their contents, including the protein myoglobin. If large amounts of myoglobin end up in the urine myoglobinuria, it can actually damage the kidneys and lead to renal failure. Clinicians can detect this using specific immunoassays for myoglobin in urine.
It's incredible how one molecule, creatinine, tells us so much about kidney function and how muscle health is linked to kidney health, too. Okay, let's shift to the dynamic fuel use in action, starting with the heart. It never stops, so what's its preferred fuel strategy?
The heart is a true metabolic omnivore, but it definitely has preferences. Under normal resting conditions, its primary fuel is fatty acids. They account for maybe 60% to 80% of its energy needs.
Wow, mostly fat.
Mostly fat. Then it uses lactate, which it can efficiently convert back to pyruvate, and glucose. Almost all of its ATP comes from oxidative metabolism. It's highly aerobic. An interesting point is that the heart prefers fatty acids even over ketone bodies, effectively sparing those ketones for the brain, especially during fasting or starvation.
Smart. But what happens when things go wrong? Like during a heart attack, ischemia.
During ischemic conditions, when blood flow and oxygen supply are drastically reduced, the heart is forced to rely more on anaerobic glycolysis. But this isn't sustainable, and it produces lactate and protons, which actually harm the heart muscle cells, lowering the pH and impairing function.
And then there's reperfusion injury.
Yes, that's a paradoxical problem. When blood flow is restored reperfusion, you'd think that's purely good news. But a sudden, rapid flood of oxygen can lead to very high rates of fatty acid oxidation. This can overwhelm the system, leading to an accumulation of NADH, which inhibits key enzymes, and paradoxically increases lactate production, further compromising the already injured cell's ability to recover and contract properly.
So fixing the blockage isn't the whole story.
Not entirely. The way the metabolism restarts matters. This understanding led to a clever clinical strategy, using drugs called PFOX inhibitors, partial fatty acid oxidation inhibitors, like trimetazidine.
What do?
They essentially dial down that excessive, potentially harmful fatty acid oxidation right after an ischemic event. This allows the heart to utilize glucose more effectively through oxidation, reducing the buildup of harmful lactate and protons, and ultimately helping the heart muscle recover better.
That's a fantastic example of biochemistry informing treatment. Now, staying with the heart, you mentioned the Circa pump for relaxation. Is there special regulation there too?
There is, yes. Specifically for the cardiac isoform, SERCA2a, its activity is modulated by a small protein called phospholamban, or PLN.
Phospholamban, okay.
Under resting conditions, PLN associates with SERCA2a and acts like a brake. It reduces its calcium pumping activity. This effectively increases the time it takes for the heart muscle to relax between beats.
So it slows things down slightly.
Right. But when you need your heart rate to increase, like during exercise or stress, your body releases epinephrine, adrenaline.
Fight or flight.
Exactly. Epinephrine activates a signaling pathway involving protein kinase A, or PKA. PKA then phosphorylates phospholamban. When PLN is phosphorylated, it dissociates from SERCA2a, or at least its inhibitory effect is greatly reduced.
Releasing the brake on the pump.
Releasing the brake. SERCA2a can now pump calcium back into the sarcoplasmic reticulum much faster. This leads to quicker relaxation, allowing the heart to beat more frequently, a faster heart rate. It's a key part of how adrenaline speeds up your heart.
And if that goes wrong?
Well, mutations in the PLN gene that disrupt this regulation can cause serious problems, like dilated cardiomyopathy. If PLN constantly inhibits SERCA2a too much, calcium isn't cleared properly, the heart muscle can't relax efficiently, the chambers enlarge, and pumping function deteriorates, leading to heart failure.
Incredible how one small protein interaction controls the rhythm of the heart. Okay, let's switch back to skeletal muscle. How does it decide what fuel to burn, say, when you're just chilling on the couch versus going for a run?
Skeletal muscle is incredibly flexible. At rest, it's pretty opportunistic. It will use whatever fuels are readily available from the bloodstream glucose, amino acids, and fatty acids. There's that regulatory interplay we talked about with ACC2 and malonyl-CoA. If energy levels are high, signaled perhaps by high citrate levels coming from the mitochondria, ACC2 is active, malonyl-CoA is produced, and fatty acid oxidation is somewhat inhibited, favoring glucose use if insulin is present.
Okay, just cruising along. What about during starvation?
During starvation, the whole metabolic picture shifts. Insulin levels are low, so glucose uptake by muscle decreases significantly. This is crucial for sparing that limited glucose for the brain and red blood cells, which absolutely depend on it.
Muscle steps back.
Muscle steps back from glucose. Fatty acids released from adipose tissue become the primary fuel, even preferred over ketone bodies, which the brain will also use. AMPK activity is high in muscle during starvation, ensuring ACC2 is inhibited, and malonyl-CoA decarboxylase is active, maximizing the muscle's ability to burn those fatty acids via CPTI.
Makes sense.
Hmm.
And then exercise.
Mm.
Huge energy demand, right?
Huge. ATP demand can skyrocket up to 100 times the resting rate almost instantly.
Wow. How does it cope?
It uses a layered approach. For the very first second or two, it uses the tiny amount of pre-existing ATP. For the next, say, 5 to 10 seconds, that creatine phosphate system kicks in, rapidly regenerating ATP.
Quick charge.
The quick charge gets you started, but that runs out fast. So almost immediately at the onset of intense exercise or during high-intensity bursts, especially in those fast-twitch fibers, anaerobic glycolysis takes over.
Glycogen to lactate.
Exactly. The muscle rapidly breaks down its stored glycogen into glucose, runs it through glycolysis without needing oxygen, and produces ATP quickly, along with lactate. This is vital because oxygen delivery from the blood can't ramp up instantly to meet the sudden demand.
That's the burn you feel in sprints.
That's largely it. Muscle glycogen stores can be depleted surprisingly quickly during intense anaerobic work, maybe just a couple of minutes. The buildup of lactate and protons lowers the pH, contributing to fatigue. This glycogen breakdown, glycogenolysis, is tightly regulated. Low ATP leads to high AMP, which activates key enzymes like PFK1 in glycolysis and also glycogen phosphorylase B, the enzyme that starts glycogen breakdown. Plus, the calcium released for contraction and circulating epinephrine also strongly stimulate glycogenolysis.
What happens to all that lactate?
It's not just waste. Lactate leaves the exercising muscle cells and can be taken up by other tissues. Resting muscles or the heart muscle can take up lactate, convert it back to pyruvate, and burn it aerobically for energy. Or lactate can travel back to the liver, where it's used to synthesize new glucose via gluconeogenesis. That's the Cori cycle.
Recycling. Okay, what about longer, less intense exercise, like jogging?
For mild to moderate intensity, longer duration exercise, aerobic metabolism becomes the dominant player.
Using oxygen.
Using oxygen. Initially, the muscle uses blood glucose, which is replenished by the liver breaking down its own glycogen stores and also making new glucose from lactate, amino acids, and glycerol. Hormones like epinephrine and glucagon stimulate this liver output. Also, muscle glucose uptake is enhanced because exercise itself, partly via AMPK, helps move GLUT4 transporters to the muscle cell surface.
So glucose is important early on.
Yes, but as the exercise continues for longer periods, say beyond 20-30 minutes, there's a gradual shift. Free fatty acids, released from adipose tissue stores, become the predominant fuel source for the working muscle.
Why the switch?
Several reasons. Hormone levels, like lower insulin, higher epinephrine, promote fat release from adipose tissue, increasing their availability in the blood. Also, the byproducts of fatty acid oxidation inside the muscle cell can actually inhibit enzymes involved in glucose metabolism. And importantly, AMPK activation during sustained exercise keeps ACC2 inhibited, ensuring CPTI is active and fatty acids can readily enter the mitochondria to be burned. The body essentially tries to spare glucose and rely more on its abundant fat stores for endurance.
Fascinating shift. Are other fuels used?
Yes, muscle can also oxidize branched-chain amino acids to some extent, especially during very prolonged exercise. It can also use ketone bodies if they're available, though fatty acids are preferred. There's also the purine nucleotide cycle, which helps buffer protons and replenish intermediates for the citric acid cycle. And even acetate, which might come from diet or alcohol metabolism, can be used as fuel by muscle.
So a very adaptable system. What about people who train regularly? How does their body change to handle all this better?
Training induces significant metabolic adaptations. Endurance training, think running, swimming, cycling, increases the muscle's capacity for aerobic metabolism. You get more and larger mitochondria, increased levels of oxidative enzymes, and enhanced ability to store glycogen and utilize fatty acids. The muscle essentially becomes much more efficient at producing ATP aerobically.
Better endurance.
Better endurance, exactly. Resistance training, on the other hand, like weightlifting, primarily leads to muscle hypertrophy. The muscle fibers get bigger. This involves complex signaling pathways that increase protein synthesis and decrease protein breakdown, leading to stronger muscles capable of generating more force.
Wow, what an incredible journey through the inner workings of our muscles.
Yeah.
From that quiet efficiency at rest to the explosive demands of a sprint, it's amazing how these intricate dynamic systems work.
It really is a biochemical marvel.
So, to kind of wrap up the main takeaways.
Well, I think we've seen that muscles aren't all the same. The different types have very specific metabolic tricks up their sleeves. Contraction itself is this incredibly fine-tuned dance involving nerves, calcium, ATP, and specific proteins.
Right.
And fuel use isn't static. It's highly regulated, constantly shifting based on what you're doing, what you've eaten, even hormone levels. The creatine phosphate system acts as that crucial rapid energy buffer.
And the clinical links are clear.
Absolutely. Understanding these pathways is directly relevant to conditions ranging from muscular dystrophies to heart attacks, kidney function assessment, and even metabolic adaptations to exercise and disease.
It really makes you appreciate all the invisible work happening just beneath the surface, making every single movement possible. Next time you reach for something or even just sit still, maybe give a little nod to that biochemical ingenuity inside you.
Indeed. And maybe here's a final thought to ponder. Considering how precisely our muscles adapt their fuel sources switching between glucose, fat, lactate, depending on the situation, what might be the really long-term metabolic consequences of a primarily sedentary lifestyle versus consistent, varied exercise on the health and flexibility of these sophisticated systems over a lifetime?
That's a great question to leave us with, something definitely worth thinking about. Thank you for joining us for this deep dive into the fascinating world of muscle metabolism.